Article 1. Revolutionary molecular device unleashes potential for targeted drug delivery and self-healing materials in a new breakthrough that could revolutionize medical and material engineering. Scientists have developed a first-of-its-kind molecular device that controls the release of researchers from the University
small molecules
of
using
Manchester
force.
describe
The
a force
researchers
control
from the University
release
of
system that
Manchester
harnesses natural
describe a force
forces
control
to trigger targeted release of molecules,
natural
which could
forces
significantly
to trigger
advance
targeted
medical
release of
treatment and
molecules,
smart
which
materials.
could significantly
The
advance
discovery,
medical
published
treatment
today in
and
the
smart
journal
materials.
Nature.
The discovery,
uses a novel
published
technique
today
using
in the
a type
journal
of
Nature.
interlocked
Uses a
molecule
novel
known as
technique using a
rotaxane.
type of
Interlocked molecule known as rotaxane. Under the influence of mechanical force such as that observed at an injured or damaged site, this component triggers the release of functional molecules, like medicines or healing agents, to precisely target the area in need, for example, the site of a tumor. It also holds promise for self-healing materials that can repair themselves in situ when damaged, prolonging the lifespan of these materials. For example, a scratch on a phone screen. Guillaume Debeau, professor of organic chemistry at the University of Manchester, said forces are ubiquitous in nature and play pivotal roles in various processes. Our aim was to exploit these forces for transformative applications, particularly in material durability and drug delivery. Although this is only a proof of concept design, we believe that our rotaxane-based approach holds immense potential with far-reaching applications. Traditionally, the controlled release of molecules with force has presented challenges in releasing more than one molecule at once, usually operating through a molecular tug-of-war game where two polymers pull at either side to release a single molecule. The new approach involves two polymer chains attached to a central ring-like structure that slide along an axle supporting the cargo, effectively releasing multiple cargo molecules. In response to force application, the scientists demonstrated the release of up to 5 molecules simultaneously, with the possibility of releasing more, overcoming previous limitations. The breakthrough marks the first time scientists have been able to demonstrate the ability to release more than one component, making it one of the most efficient release systems to date. The researchers also show versatility of the model by using different types of molecules, including drug compounds, fluorescent markers, catalysts, and monomers, revealing the potential for a wealth of future applications. Looking ahead, the researchers aim to delve deeper into self-healing applications, exploring whether two different types of molecules can be released at the same time. For example, the integration of monomers and catalysts could enable polymerization at the site of damage, creating an integrated self-healing system within materials. Article 2. Swimming in schools makes fish surprisingly stealthy underwater, with the group able to sound like a single fish. The new findings by Johns Hopkins University engineers working with a high-tech simulation of schooling mackerel offers new insight into why fish swim in schools and promise for the design and operation of much quieter submarines and autonomous undersea vehicles. It's widely known that swimming in groups provides fish with added protection from predators, but we questioned whether it also contributes to reducing their noise, said senior author Rajat Mittal. Our results suggest that the substantial decrease in their acoustic signature when swimming in groups compared to solo swimming may indeed be another factor driving the formation of fish schools. The team created a 3D model based on the common mackerel to simulate different numbers of fish swimming, changing up their formations, how close they swam to one another, and the degrees to which their movement cinched.
The model, which applies to many fish species, simulates 1 to 9 mackerel being propelled forward by their tail fins. The team found that a school of fish moving together in just the right way was stunningly effective at noise reduction. A school of 7 fish sounded like a single fish. A predator, such as a shark, may perceive it as hearing a lone fish instead of a group. Asterisk, Mittal said. This could have significant implications for prey fish. Asterisk, the single biggest key to sound reduction, the team found, was the synchronization of the school's tail flapping, or actually the lack thereof. If fish moved in unison, flapping their tail fins at the same time, the sound added up, and there was no reduction in total sound. But if they alternated tail flaps, the fish canceled out each other's sound. The researchers found sound is a wave, Mittal said. Two waves can either add up if they are exactly in phase, or they can cancel each other if they are exactly out of phase. That's kind of what's happening here, though we're talking about faint sounds that would barely be audible to a human. The tail fin movements that reduce sound also generate flow interaction between the fish that allow the fish to swim faster while using less energy, said lead author Joe, a Johns Hopkins graduate student studying mechanical engineering. We find that reduction in flow generated noise does not have to come at the expense of performance, Joe said. We found cases where significant reductions in noise are accompanied by noticeable increases in per capita thrust due to the hydrodynamic interactions between the swimmers. The team was surprised to find that the sound reduction benefits kick in as soon as one swimming fish joins another. Noise reduction grows as more fish join a school, but the team expects the benefits to cap off at some point. Simply being together and swimming in any manner contributes to reducing the sound signature, Mittal said. No coordination between the fish is required. Next, the team plans to add ocean turbulence into the models and create simulations that allow the fish to swim more freely.
Article 3.
Pregnancy may carry a cost, reports a new study. The research shows that women who reported having been pregnant looked biologically older than women who had never been pregnant, and women who had been pregnant more often looked biologically older than those who reported fewer pregnancies.
This study builds on epidemiological findings that high fertility can have negative side effects on women's health and longevity. What was unknown, however, was whether the costs of reproduction were present earlier in life, before disease and age-related decline start to become apparent. Until now, one of the challenges has been quantifying biological aging among the young. This challenge was overcome by using a collection of new tools that use DNA methylation DNAM to study different facets of cellular aging, health, and mortality risk. These tools, called epigenetic clocks, allow researchers to study aging earlier in life, filling a key gap in the study of biological aging. Epigenetic clocks have revolutionized how we study biological aging across the life course and open up new opportunities to study how and when long-term health costs of reproduction and other life events take hold, said Kaylin Ryan, PAAHDAADDDDHEHE, lead author of the study and associate research scientist in the Columbia Aging Center. Our findings suggest that pregnancy speeds up biological aging, and that these effects are apparent in young, high-fertility women, said Ryan. Our results are also the first to follow the same women through time, linking changes in each woman's pregnancy number to changes in her biological age. The relationship between pregnancy history and biological age persisted even after taking into account various other factors tied to biological aging, such as socioeconomic status, smoking, and genetic variation, but were not present among men from the same sample. Despite the striking nature of the findings, Ryan encourages readers to remember the context. Many of the reported pregnancies in our baseline measure occurred during late adolescence, when women are still growing. We expect this kind of pregnancy to be particularly challenging for a growing mother, especially if her access to health care, resources, or other forms of support is limited. Ryan also acknowledged that there is more work to do. We still have a lot to learn about the role of pregnancy and other aspects of reproduction in the aging process. We also do not know the extent to which accelerated epigenetic aging in these particular individuals will manifest as poor health or mortality decades later in life. Ultimately, I think our findings highlight the potential long-term impacts of pregnancy on women's health and the importance of taking care of new parents, especially young mothers.
Article 4. New hypothesis paper builds on a growing scientific consensus that Parkinson's disease route to the brain starts in either the nose or the gut, and proposes that environmental toxicants are the likely source. A new hypothesis paper appearing in the Journal of Parkinson's Disease on World Parkinson's Day unites the brain and body, first models with some of the likely causes of the disease environmental toxicants that are either inhaled or ingested. The authors of the new study, who include Borgammer, argue that inhalation of certain pesticides, common dry cleaning chemicals, and air pollution predispose to a brain-first model of the disease. Other ingested toxicants, such as tainted food and contaminated drinking water, lead to body-first model of the disease. In both the brain-first and body-first scenarios, the pathology arises in structures in the body closely connected to the outside world, said Ray Dorsey. Here we propose that Parkinson's is all-systemic disease and that its initial roots likely begin in the nose and in the gut and are tied to environmental factors increasingly recognized as major contributors, if not causes, of the disease. This further reinforces the idea that Parkinson's, the world's fastest-growing brain disease, may be fueled by toxicants and is, therefore, largely preventable. Different pathways to the brain, different forms of disease. A misfolded protein called alpha-synuclein has been in scientists' sights for the last 25 years as one of the driving forces behind Parkinson's. Over time, the protein accumulates in the brain in clumps, called Lewy bodies, and causes progressive dysfunction and death of many types of nerve cells, including those in the dopamine-producing regions of the brain that control motor function. When first proposed, Brock thought that an unidentified pathogen, such as a virus, may be responsible for the disease. The nose and the gut are lined with a soft, permeable tissue, and both have well-established connections to the brain. In the brain-first model, the chemicals are inhaled and may enter the brain via the nerve responsible for smell. From the brain's smell center, alpha-synuclein spreads to other parts of the brain, principally on one side, including regions with concentrations of dopamine-producing neurons. The death of these cells is a hallmark of Parkinson's disease. The disease may cause asymmetric tremor and slowness in movement, and a slower rate of progression after diagnosis, and only much later, significant cognitive impairment or dementia.
When ingested, the chemicals pass through the lining of the gastrointestinal tract. Initial alpha-synuclein pathology may begin in the gut's own nervous system, from where it can spread to both sides of the brain and spinal cord. This body-first pathway is often associated with Lewy body dementia, a disease in the same family as Parkinson's, which is characterized by early constipation and sleep disturbance.